Hello everyone, how are you all? Welcome to One Page Biology. This is Praful. I hope you all are doing fine. In today's video, dear students, I am going to help you understand a very important topic and a concept which is called as recombinant DNA technology. So dear students, this topic is part of the chapter Biotechnology, Principles and Processes. So are you all ready for this topic? Let's begin with the video, dear students. So what do we understand by the process of recombinant DNA technology? So dear students, as you all can see in this particular diagram, there are two pictures. One is of a bacteria, another is of a human cell. So suppose dear students, if we want to form many copies of a particular gene which is present in the human DNA. Let's say a gene is basically coding for a hormone which is insulin hormone. How is that possible? So the answer to this question lies in the concept of recombinant DNA technology. As you all can see in the diagram of a bacteria, there is one extracircular DNA. What is this extracircular DNA called? This extracircular DNA is known as a plasmid. And inside the plasmid, can you all see there is some red color gene segment. What is that red color gene segment? Yes, dear students, that gene is nothing but the gene which is called as a desired gene. That is the gene of which we are going to form many copies. So this technique of forming many copies of a particular gene of interest by using the plasmid of a particular bacterial cell is known as recombinant DNA technology. So dear students, who invented this particular method? Now back in the year 1972, dear students, there were two scientists. One was Herbert Boyer and the other was Stanley Cohen. So what did they do? So they were the first ones to actually make the first artificial cloning vector. So that first artificial cloning vector was basically formed by using the plasmid of a bacteria which was called as Salmonella typhimurium. Yes, dear students, that is the same bacteria which causes the disease which is known as typhoid. So by using the plasmid of Salmonella typhimurium, Stanley Cohen and Herbert Boyer had formed many copies of a particular gene of interest which was mainly coding for antibiotic resistance. So let us understand this particular technique of recombinant DNA technology. Now as you all can see in this particular picture, dear students, there is a particular vector which is called as a plasmid vector. Now what is exactly a plasmid vector and why is it called as a vector? First of all, as we just discussed in the earlier slide, a plasmid is nothing but an extra chromosomal DNA which is present in the bacteria. Now you must have often heard about certain bacteria which are able to survive at high temperatures or which are basically antibiotic resistant. So from where do the bacteria get the power to resist the antibiotics or even to survive at high temperatures? So dear students, this power in the bacteria comes from the plasmid. Yes, dear students, the plasmid contain some antibiotic resistant genes or certain genes which give them the ability to survive at high temperatures. So today in the recombinant DNA technology, the same plasmid, which is a extra chromosomal DNA, can be used also to form many copies of a particular gene. Now dear students, getting into the technique part of the recombinant DNA technology, what is exactly done is basically the plasmid, which is the extracellular DNA, is combined with the gene of interest. Now the question is, how is this possible? Of course, for that, the plasmid has to be cut at specific sites. So how can we cut the plasmid at specific sites? So dear students, for cutting the plasmid at specific sites, there is a role of some very important enzymes. These enzymes are called as restriction enzymes. So dear students, the restriction enzymes are special class of enzymes which are capable of cutting the DNA at specific sites. Now once the plasmid has been cut at a particular site with the help of a particular restriction enzyme, now it is possible to fix a desired gene segment inside that particular site. Even the source DNA has to be cut into many fragments so that we can get the desired gene from the source DNA. So for example, if we want a human insulin gene, so we have to cut the human DNA which can be obtained from the pancreatic cells of humans. So the human DNA from the pancreatic cells will be cut into many fragments, and from that the desired gene of interest, that is the insulin gene, is basically selected. So this is essentially performed by the restriction enzymes, dear students. So once the desired gene and the plasmid has been cut at specific sites with the help of restriction enzymes, these two are combined. So how are they combined? Now for combining them, there is a special class of enzyme which is used, and this is known as DNA ligase. So dear students, DNA ligase enzyme helps in joining the plasmid and the desired DNA fragment. Once the plasmid and the desired gene has been ligated or connected, such a DNA is called as recombinant DNA. So dear students, this recombinant DNA is now ready to be transferred into a host. Now what is a host, dear students? Most of the times the host which is being used is nothing but a bacteria. So very often E. coli is used to transfer the recombinant DNA because a bacteria has a high power of multiplication. So as the bacteria multiplies rapidly, even our recombinant DNA is going to multiply, and because of that we are going to get many copies of our desired gene. So today dear students, there are many applications of recombinant DNA technology. Say for example, a gene which is coding for vitamin A has to be transferred into a particular plant. So how are we going to transfer that particular gene of vitamin A into a particular plant? So for that we are going to use a plasmid which is known as TI plasmid. Dear students, this particular plasmid, TI plasmid, stands for tumor inducing plasmid, and this plasmid is obtained from a bacteria which is known as Agrobacterium tumefaciens. So inside this TI plasmid, the desired gene of interest, that is the gene which is coding for vitamin A, is fixed. So now this recombinant DNA is transferred inside a plant cell which is possibly a meristematic cell. These meristematic cells are now transferred inside the plant. So now the plant has got the cells which has got the gene which is coding for vitamin A. So this particular plant, when it is going to grow and ultimately form a particular fruit, it is going to be rich in vitamin A. So this way, dear students, today it is possible to produce plants with the desired characteristic. Let us discuss another important application of recombinant DNA technology. So suppose a particular organ has got cancerous cells at an initial stage. So dear students, these cells can be possibly removed from that particular organ. Once these cancerous cells are removed, dear students, inside the DNA of these cancerous cells, the desired gene of interest, which can be a therapeutic gene, is inserted. So these therapeutic genes can inactivate the cancer cell genes. This way, dear students, it is possible to stop the multiplication of cancer cells inside the humans. So like this, there are many more applications of recombinant DNA technology. If you know any other applications of recombinant DNA technology, you can definitely mention them in the comment section. If you have understood this concept of recombinant DNA technology, do like the video, dear students. Also share with your friends, and please don't forget to subscribe the channel One Page Biology. If you have any doubts or suggestions, do mention in the comment section. See you all in the next video, dear students, with some another biology related concept. Till that time, take care of yourself. Thank you so much. This is Praful signing off. Take care. Bye bye.